so this paper, it came out in Cell, and it came out, let's see if I can remember the date. Well, December 21st. This paper came out in the future. December 21st, 2023. Okay, so this paper is in the future. This is from the future. We are reading a paper from the future. And it is about choroid plexus mast cells drive tumor-associated hydrocephalus. So this is in the context of a tumor in the brain, specifically like a metastasis. And let's just look at this fun little diagram they have here. So here, I'm outlining in pink the ventricles in the brain. And then the choroid plexus is that fuzzy fun stuff, and I'm going to do that in blue. This is the choroid plexus, okay? It looks like a sea anemone or a branch. Basically, it's a bunch of epithelial cells. So epithelial cells are like, you could think of them as skin cells almost. Skin cells are a type of epithelial cell. Choroid plexus. And then they're going to be talking about the little mast cells. So we'll draw mast cells in green. The mast cells are those exploding immune cells. Now, they're not necessarily exploding. The cell is still intact, but they release granules rapidly when they become activated. And we'll talk a little bit about how mast cells typically work in allergy, although these mast cells are very different from the mast cells that occur during allergy. These mast cells lack a very crucial receptor that's only found on the mast cells that react to allergens. And that receptor it recognizes IgE, which is a type of antibody that's released during allergy. But these mast cells don't have that receptor. So they're a whole new class of mast cells. So this paper not only identified the mechanism by which these mast cells caused the swelling to occur, but they also identified these mast cells as being a totally new type of mast cell, which is really cool. Okay, so the authors of this paper, the first author is Ye Li. Usually the first author is typically the one who does the most work in terms of like all the experiments and like the intellectual ideas. And then usually their advisor will be one of the last authors, like maybe Xisheng Su or something. I've never heard of this lab or these researchers, but there's many researchers out there. So, and I also am not in the brain tumor space. So this is kind of outside of my niche. Okay, so I'm just going to read off some of the highlights from the paper just to orient you guys. Also, if you are just joining and you're curious about this paper, you can find it in my Discord under the Nerdy Things channel. So there you go. So first of all, they found that these choroid plexus mast cells increased during tumor-associated hydrocephalus. So this is when there were brain metastases or tumor metastases in the brain, and also in conditions of hydrocephalus, which again is swelling. The mast cells impair the choroid plexus epithelial cilia and cause the tumor-associated, or TAH. We'll call it TAH, but I'll call it hydrocephalus because acronyms suck. So the mast cells impair the choroid plexus epithelia cilia and cause hydrocephalus via tryptase. Tryptase is an enzyme. I believe it cleaves tryptophan, maybe. I don't know too much about it. They don't go too much into what it's actually cleaving, but there is a receptor that tryptase binds to that results in the impaired epithelial cilia. Now, if you're wondering what a cilia is, a cilia are these little fingers on the surface of the epithelial cell. So these little fingers, these are cilia. And then the receptor that they're looking at is PAR2. That's the receptor that's going to bind to tryptase, which is released by the mast cells. So they say that the tryptase in human CSF was associated with clinical severity of hydrocephalus. So then after they studied this in mice, they went back to humans and they looked for tryptase in human CSF, or cerebral spinal fluid, and they found that the more tryptase there was in human cerebral spinal fluid, the more severe their hydrocephalus was as well. So there's a strong correlation between those two things. And then they also were able to use a drug to actually target tryptase and block it from binding to PAR2. And they found that this reduced the symptoms in mice. They haven't tested this in humans because the drug actually also increases hemorrhaging, which you don't want to have hemorrhaging in the brain. It isn't too severe, but they were like, yeah, if we are going to do this in humans, we need to find like a different type of this drug that doesn't also cause hemorrhaging, because brain hemorrhaging is not something you want to have after you've just recovered from hydrocephalus. I guess we should talk a little bit about what mast cells are. So they're immune cells in your body. And the thing is, you don't want a lot of immune cells in your brain doing crazy things, because they'll cause swelling in your brain. And the swelling in your brain is called hydrocephalus. This usually happens so people can not spontaneously, but they can get hydrocephalus for other reasons, but we're going to be talking about it in the context of cancer. So sometimes people get brain metastases. So some cancer breaks off, like the little cancer cells go into the bloodstream, they go up past the blood-brain barrier, and then they form little tumors in the brain. And what can happen is that the brain tumor, could get really big, and then it can cause pressure to occur inside of the brain, and the pressure pushing on different parts of your brain can result in death. There's a second way, though, where instead of the tumor just getting big, because people who have a big tumor, usually they get it resected. Obviously, you don't want to go around with a tumor in your head, right? That would be kind of annoying. So people get the tumor removed, but they still have this pressure in the brain, this hydrocephalus. But what is causing it is a buildup of liquid called CSF, or cerebral spinal fluid. And the cerebral spinal fluid typically bathes your entire brain, and it also is found in your spine, obviously, spinal fluid. If you've ever had a spinal tap, they're taking out some of your cerebral spinal fluid, or CSF. So the thing is if you have too much in your brain, especially in like your ventricles, it'll cause your brain to swell, and people die from this condition as well. So too much liquid in the brain is bad. And we're going to be talking about how mast cells actually increase the amount of this liquid in the brain. And we're going to go through the entire mechanism. It's going to be so much fun. Okay, so I love that little brain thing. Okay, so on the right, or on the left side, we're looking at a sort of 3D. These are false colors, the blue and yellow, that they have added for contrast. And on the right side, what we're looking at is we're looking at kind of a top-down view of the mast cells. There are these big kind of bulby cells. Now the little tiny cells up above, I'm not actually sure if those are mast cells, but the big flat pancake looking thing is definitely a mast cell. And the mast cells are doing this thing called degranulation, which literally means they're releasing granules into, the abyss. Let's look at that again. So first you'll notice there's no dots, and then you'll start to see these little white dots popping up. And those are just like granules that are being shot out of the mast cell. And these granules, they are not just, they're active. So they contain different things like that basically kill other cells. The other thing I wanted to kind of orient you guys with was where in the brain we are located. So the structure I just want you guys to focus on is the blue structure. Those are the ventricles. That's a hollow space, and it's filled with cerebral spinal fluid. And when the ventricles get too full of that cerebral spinal fluid, or CSF, it causes pressure. It pushes against other really critical brain structures, and that can result in pressing on blood vessels. So then you're starving the brain of oxygen. Like certain components of the brain won't get oxygen because you're squishing the blood vessels. And also the pressure, I believe, can also somehow result in death, which I honestly, other than the blood vessels getting squished, I'm not sure how the pressure is actually killing the brain. But anyways, so these ventricles, we're going to be talking a lot about them because in the study, they had a model in mice where the brains of the mice, their ventricles were getting filled with lots of cerebral spinal fluid and then causing the mice to die and be really sick. And this was kind of independent of the tumor bulk. So the tumor itself was really small, but there was still a lot of pressure. And they wanted to know, okay, if the tumor is not causing the pressure, what is making these ventricles swell? And they discovered mast cells. So we're going to get into a little bit of the background. So first of all, this is taking place in patients with metastases, which are just tumors, so basically tumor cells that have broken off from the main tumor in another organ in the body and then have traveled to the brain, seated themselves in the brain, and caused a secondary or tertiary tumor there. So here's some stats. Recent epidemiologic studies have shown that brain metastases afflict approximately 20 to 30% of patients with cancer. That is a pretty big number. And we're not even talking about patients with primary brain cancers like glioblastoma. These are just people who had cancers somewhere else, like melanoma, for example. Melanoma is a skin cancer, but you can end up with complications if the melanoma cells travel to the brain and cause brain mets. So unfortunately, the prognosis remains notoriously dismal. So that means if you are diagnosed with brain metastases, unfortunately, it's a dire situation for you. So anyways, intracranial hypertension, so this is basically swelling inside of the brain, is one of the leading causes of death in patients with brain tumors. Tumor-associated hydrocephalus, or TAH, which is what the paper will call it, we'll just say hydrocephalus, is a common complication of intracranial malignancies, such as brain cancers. The incidence of TAH, or hydrocephalus, can be up to 90% in patients with deep midline brain tumors. So conventional wisdom, so what we think is happening is that the tumor compression blocks the flow of cerebral spinal fluid, kind of like that cyst, and results in hydrocephalus. However, there's another type of hydrocephalus called communicating hydrocephalus, and these are often present in cancer patients. So what does that mean? So communicating hydrocephalus, I looked this up because I wasn't sure, and I'm going to just reread the definition because it was really weird. I'd never heard of it. It occurs when the flow of CSF is blocked after it exits the ventricles. So the ventricles themselves are still like open. Those aqueducts that we saw in the video, those are still open, but somehow the flow of CSF is blocked, maybe leaving into the spine. Okay, so some of the reasons why we don't understand communicating hydrocephalus is because there aren't that many preclinical models. So there aren't enough animal models to study this, or there aren't really any good animal models to study this. And so that's actually one of the things this paper does first, is they develop a model of communicating hydrocephalus. So they need to create a mouse model. So what they did first is they used a mouse breast cancer cell line, and they also used a Lewis lung carcinoma cell line, and they injected these into the internal carotid artery of mice. So why did they use the breast cancer and lung cancer models? I am actually not sure. I'm guessing that these might be cancers that have a lot of metastases, and they know that they can metastasize and survive in the brain. They injected them into the artery of the mice because the blood flow, if you basically remember how I talked about metastases, they're basically little tumor cells that have broken off the main tumor and they get into the blood, and then the blood obviously goes into the brain. And so this is a great way to introduce cancer into the brain. Just put it into an artery, put the cancer cells into an artery. Eventually, they're going to get up to the brain. Freezes. You found your favorite alert. Freezes, welcome in. How are you? Going back to this, what was I talking about? Right. So they have they introduce these mouse cancer cells into the mice through the artery. They travel up to the brain and they form tumors in the brain. So what they do is they grow up the tumors, and then they harvest the tumors from the mice that develop ventriculomegaly. So what this means is that the ventricles have gotten big. Megaly means like large. So these are enlarged ventricles. And if you'll remember, the ventricles are that space inside the brain that fill up with fluid. So they take tumors out of the mice that develop this, and then they re-inject them into the internal carotid artery. They do this whole process again. They also, so one thing that they made a note of is that mice with brain macrometastases, so these are really big tumors that literally physically press against the brain, they do not include those because, again, remember there's two different types of hydrocephalus. One where the tumor is really big and pressing against the brain, and then the communicating hydrocephalus where the ventricles swell up. So they're only selecting tumors that cause ventricles that swell up. So they do this like 3 or 4 times, and there's a fun little picture, I think, that we can look at where they kind of diagram how this happens. So here they are putting the cancer cells into an artery, and then the cancer cells go and create a tumor. And then here they have, in this case, the tumor is small, but it causes the ventricles to swell. They take that cancer out, they expand it, they re-inject it into another mouse, and then they do this until they get, actually, I think, yeah, let me see, why did they do it 4 times? I guess they wanted to purify them or make sure that they were really looking for cancer cells that somehow, something about the cancer cells, they maybe release like protein cues that somehow will cause this swelling of the ventricles. And they don't know what that is, which is why they're doing it this way. It seems very sort of like you're shooting something into the dark where you're selecting for a cancer that does the phenotype that you want, but you don't really know how it's doing that. But they have to do that if they don't know the cause. So anyways, so now that they have selected for these tumors that cause the swelling, they actually want to image and show that the swelling is happening. So in this image here, what we're looking at is a special type of microscopy that forms a 3D image of the brain. And let me use my hot pink color. These are the ventricles in the orange. These fiery things, those are ventricles. So this is a normal mouse. This mouse does not have cancer, and you can see the ventricles are nice and small and beautiful. They also have a cross section down here, which you can't really see, but here you could see that the ventricles are nice and normal sized. Then they put the parental version of the breast cancer. This is the unselected cancer. So this is a mix of different cells. It might not cause the swelling that they expect. And you can see like the ventricles, they look kind of like the normal ones, right? But maybe slightly bigger. And then they also, remember when they were doing this selection, there were mice that never developed swelling ventricles. And these are the mice that had the tumors that did not cause the swelling ventricles. So these are kind of like a negative control. So you can see the ventricles are still really small. And then they have their interest group, right? This is the cancer that caused ventricle swelling. So these ventricles are supposedly bigger. You can actually see it a lot better if we look down at this cutaway. You can see that the ventricles are really, really big. And yes, I'm aware this looks like a phallic object, but just get your mind out of the gutter, okay? So these ventricles are much bigger than the negative control, where the ventricles are really skinny and small. So here, there's more fluid, more swelling, right? So they establish their model. So then they actually measure the amount, like the volume or the space within the ventricles, and they quantify that. So remember, E0771 is our breast cancer, and then VM down here is our ventricular megaly condition. So VM from now on, just think of that as big ventricles. If it makes it helpful, you can think of it as like this weird, large, phallic-shaped object. This is the VM. These are the really swollen, large ventricles filled with lots of liquid. And so here they're actually quantifying that increase in size, and you can see in the red bar that the size increases a lot. So on the y-axis, they're looking at the volume in millimeters cubed, and compared to like normal mice, these are normal mice, the ventricle size is really large. And then they do this in 2 other types of cancers, and they see the same thing. So they've done the same selection process. But anyways, they see in the VM condition with the swollen ventricles that there is indeed an increased volume. Then they also look at this interesting thing where they're looking at pressure on the eyeballs. I've never seen this test before, so I don't really know what I'm looking at, but these are supposedly retinas of the mice, and this is the condition with the swelling, and I'm not sure, I guess like there's less blood flow in a certain area, because I guess this is blood flow. I didn't really know what this figure was showing, so we're going to skip that. I also didn't know what this was, so we're also going to skip this one. These are like neuroscience, like surgery type tests and assays that they did. Then what they do is they actually stick a catheter into the brain of the mouse, and they have a little device that measures the intracranial pressure, so the amount of pressure inside of the brain. And in the mouse with the large ventricles, as one would expect, we see a super big increase in the amount of pressure. So pressure here is measured in, oh my God, I think this is millimeters of mercury. This is just like the higher up it is, the more pressure there is. So in all 3 cancer models, you can see that there's this increase in brain pressure. So far, this is mimicking what we see in humans, right? We see that in humans who have hydrocephalus, the ventricles get bigger in volume. We saw that here, and they have more pressure in their brain. They also measure neurological scores in the condition with the enlarged ventricles. They see that the patients have a worse neurological score, and they see this. These are the mice. So the mice, they're just trying to show that the mice display similar symptoms to the humans when they have these enlarged ventricles. And then the most important metric, I think, is, well, maybe not important, but it is very, very important that we know this, but it's the overall survival. So how long did the mice survive? So the blue line here, parental, this is just the normal tumor. So you can see that with the normal tumor that hasn't undergone selection, mice typically live 50 days. This on the y-axis is the overall survival. So you can see that the first mouse dies somewhere right around here at day 40, and then you have other mice dying all the way up until day 50. This is called a survival curve. So every time, might have a cohort of 10 mice, and as each one of those mice dies, you'll make a little point on this chart. And so over time, you'll lose, you'll go from 100% of the mice surviving down to 0% of the mice surviving. And you can graph that out in this way. When we look at, and then the negative control nicely kind of copies that, where there's still pretty decent survival. But then when we look at the mice that have the really large ventricles, their survival is really bad. So these mice die around 20 days. And they measure the significance of this, and this is quite significant. So mice with the swollen ventricles in the brain live less. They die faster. They looked at survival in the 2 other types of cancer. And again, if you're looking just at the red line, which is the bad condition, the big ventricles, the survival is generally much worse than the normal tumors. So now they've successfully made a model of a very aggressive type of tumor that causes this swelling. And it's not based on the size of the tumor. It's based on just certain cues or certain aspects of the tumor that are causing some change, and they figure out what that is later on, but they've now selected for that tumor, and they've created a nice animal model to study this hydrocephalus. So what they wanted to do was see maybe these special tumors with the bigger ventricles as well, because again the ventricles are swelling because of all that fluid, maybe they're just making more cerebral spinal fluid. And guess what? They measure the amount of cerebral spinal fluid produced. They have this really interesting way of measuring it that I don't fully understand, but they're measuring microliters per minute. So I think what's happening is I think they use the oil, and then they kind of have this little glass tube, and they can measure how much of the CSF is being pushed out of the glass tube, and that is how they get this microliters per minute. I don't know. basically, the big takeaway is that they see a ton more CSF being made in their aggressive tumors. So the red bar goes up. So something is causing more CSF production in the brain, which is then causing the swelling part. So before this paper came out, and also just kind of historically in the field, what they knew is that many clinicians usually assume that this communicating type of hydrocephalus, so hydrocephalus occurring without a large tumor, results from tumor debris, so the small tumor debris, clogging up the arachnid granulations. So I think these are like little like channels inside of the brain. I don't exactly know what the arachnid granulations are, but they think that the CSF is just getting stuck because there's clogging happening. And this is because when they take samples of the CSF from patients, they see high protein levels. So then these people, they decided to check if there were indeed high protein levels, because maybe blockage is happening, even though they didn't see that yet. So they did observe elevated CSF protein levels inoculated with the mice, or inoculated with both the non-aggressive and the aggressive tumors. So remember, the VM cells are the cells that cause big ventricles, and the un-VM cells are the negative controls, or the tumors that don't cause big ventricles. And so they need that negative control kind of to compare, like, okay, here we have a normal like tumor cell that isn't causing this problem, and then we have the aggressive tumor cell that is causing the problem. But they saw that both of those tumors had more protein levels. So that really wouldn't explain, that really wouldn't support the hypothesis that the more aggressive cancer was resulting in more proteins, which would then result in clogging. So the clogging doesn't seem to be the issue. Okay, they were looking at drainage. They wanted to see if there were any differences in the drainage of the CSF, and they used this kind of label called, they used a fluorescent protein to kind of label the CSF, but they didn't see any differences in drainage either. All right, as you can see, I was prepared, and I already drew my own little brain. This is my cute brain. So what we know is that sometimes when a patient gets a brain metastasis, so this is like a brain tumor, the tumor can cause swelling independent of its size. So this tumor could be really small, but the patient will still have swelling in the brain. And the swelling is because, I don't know if this is the right color, it's because they have a lot of CSF, or cerebral spinal fluid, pooling up inside their ventricles. So the blue stuff is the CSF. They have a lot of it. And guess what this does? It pushes outwards. So we have this pressure pushing out, and this can block blood vessels and ultimately result in death if not treated. And this little structure here is called the choroid plexus. This is going to become important in like figure 2 or 3. So anyways, what they've done so far is they were like, this happens in humans, but we need to be able to study this. So we need a mouse model. It looks like Brian. So they basically were able to show that in a mouse, where they used a mouse tumor, they were able to also see that they use a little tiny tumor, but they still get this expansion of the ventricles. And the reason why, they find out that it's because the cerebral spinal fluid is being produced in vast amounts. So there's just tons and tons of the cerebral spinal fluid being produced, and it's swelling up the brain. So that's where they are right now. So if you were a scientist, what would you want to know next, right? What's triggering the CSF overproduction? That's a great question. If I know that you said that before I asked, so let's go back to the paper, because that's what they asked as well. So we go back here. The CSF is filling up the ventricular space. It's the liquid that's literally, like, imagine you have a balloon and you fill it with water. That water is the CSF, and eventually your balloon is going to get so big it's going to burst. That's what's happening here. But they want to know, okay, but why is the brain overproducing CSF? That's not normal, right? That's ultimately like what they want to know. So what they do is they decide, well, let's start by looking at all of the different cells that are located in the ventricle. Maybe one of those cells is responsible for causing the brain to make more CSF. So what they do is they do this really complicated sequencing. They do single nucleus RNA sequencing. So let me just draw a picture of what single nucleus RNA sequencing is. We have a cell, and inside of the cell, we have something called a nucleus. And a nucleus means a single nucleus. Many nucleus are called nuclei. So that's what the whole like single nuclei. It means they're measuring multiple single nucleuses. So what they did with this sequencing, they wanted to know the identity of the cell. Now, the identity of a cell is in its DNA, and its DNA is housed in the nucleus. This is my really shitty DNA molecule. That's not really what DNA looks like. Just go with it, okay? I can't draw. So here is the DNA. Now, the cool thing about DNA, let's pretend that we're unraveling the DNA. Here's a fragment of it, and here is something called a gene. A gene is literally a combination of different nucleotides, which are just like these little like proteins, sort of, or not protein, they're like these chemical structures. And basically what happens is this gene can get copied into RNA. Now, RNA is just basically the same as the DNA, but it's made out of like complementary or matching base pairs. So, for example, that's a bad color. Let's say that we have like an adenosine, a thymine, et cetera, et cetera. Adenosine, thymine, they link together and they're some of the building blocks of DNA. In the RNA, you would actually have, U. So this is the confusing thing about RNA. I believe is it A to U? Okay, well, whatever. And T to A, something like that. You get like a complementary switching of the RNA. And then this RNA eventually goes on to become protein. Oh my God. I am losing my ability to write and speak at the same time. So the RNA then becomes a protein, and the protein is like the actual functional thing. So what they did with this sequencing is they literally just took the nucleus out of a cell. They isolated it using this fancy method where they use like beads, and the beads have some stuff to it that just stick to certain things on the nucleus. And that's how you can isolate the nucleus. Basically, though, it will give the scientist information about what the cell's identity is, and that's how they identify all of the cells in the ventricles. So that was my terrible art. So they sequence all of these cells and they get this really complicated looking map right here. So let's just unpack what we're seeing other than a bunch of like paint splatter on a wall. So first of all, each dot represents one cell, presumably. So each of these, if we zoom in, I can't zoom in that far, but you can see how there's a bunch of dots. So those should represent one cell. And then they have the normal, like healthy mouse condition here in blue. And then they have their tumor-bearing mouse with the really aggressive cancer in pink. And you'll notice, so basically the way that they plot this is they plot the cells based on how many similar components they have. So cells that are more similar will cluster together, like up here. These cells all have something in common. Now, what that something is found over here. So they now look at other like traits of these cells, and they find that these are actually epithelial cells. Now, let me pick a brighter color. So these are like the skin cells almost. So the epithelial cells are located here. So in what they notice is that in the mouse with the tumor, there are more epithelial cells because only the tumor-bearing mice have these epithelial cells. That's kind of weird. Okay. They also notice that in the other cluster that we have mostly pink dots in, which is the tumor, we see that those match to immune cells. Okay, so there's more immune cells in the tumor-bearing mouse. That's not really unexpected. You would expect immune cells to be recruited to a tumor, right? Let's see what else. I think there's some other cells, but these are the main ones. Then they take it a step further and they're like, okay, so what are those immune cells? And they identify in the pink. So again, remember the pink is the tumor. They identify mast cells as being really, really enriched in the tumor-bearing mice. For some reason, in the ventricles, there's a ton of mast cells. There's also a lot of macrophages, which we know are the cells that are responsible for kind of picking up dead cells, and then they like kind of break them down, and then they present the peptides out on their surface. Macrophages also do other things. They're usually found in brain cancers because they actually can make the brain cancers. They help support the growth of brain cancers sometimes, which is really bad. But they see more macrophages, but they also see a ton of our dear friends, the mast cells, which are the cells that release granules. So already this is telling them, okay, for whatever reason, in the mice with the swollen ventricles, there are also a ton of these mast cells. So maybe the mast cells are doing something. It could be quite possible that the mast cells were not doing something, but this is just what they see. Okay, so now they're looking at where these mast cells are located. And they can do this by taking tissue sections and then looking at the RNA that's expressed in certain parts of the tissue section and making a map. So you'll see these broadly kind of look like a brain, right? Okay, so you'll see that this grid somewhat looks like a brain, and the different colors are corresponding to RNAs that are specific to these like structures. So for example, we have the cerebral cortex, we have the ventricular system, which is kind of spotty, but remember the ventricular system is the cavity, but there's also cells present there. So they can find RNA there. And most of this is really confusing to look at. So I just want to find the part where they talk about the mast cells. Where did they do that? We're going to skip all that. Okay, and we're going to go down to this map because this is actually the important one. They look for the expression of RNA transcripts that are specific to mast cells in the ventricle space, and you can see these fun little dots right here. And also this like red zone. This is where they find all of the mast cells.
Yeah, my dick's full of Windex.
Cloven Acorn, hello and welcome in. How are you? I hope you're doing well. Cloven, your package will be on the way on Monday. I have been just procrastinating like crazy. Regular Steve, thank you so much for the follow. That was very scary. All right, Regular, thank you so much. And virology, we'll do more. Okay, virology is a scary topic for me. Immunology, we can do some more of. I love immunology. Okay, so they find that, okay, all of these mast cells that they found in the brain, they're actually located in the ventricle. Wow. Okay, so this is where the swelling happens. So maybe the mast cells are doing something. They do some more imaging. So this is a fluorescent image of the inside of like the brain structure. So the choroid plexus is this little, let me find a brighter color. It's this little strip of tissue here that I'm tracing around. And then they're going to zoom in where this white box is. This white box is a zoom in, and they see one singular cell. This red cell, holy shit, freezes. This red cell is a mast cell. Freezes is being a disruptive child. So they use something called tryptase, which we're going to talk about more, but tryptase is expressed by mast cells, and they can use that to label them inside of the tissue. So then they go on and they look at other parts of the brain and they find more mast cells in red. And then the vasculature, they've just labeled blood vessels for contrast. Okay, so now they've identified mast cells as being important, so we're going to move on. Okay. Wow, what did we do here? Oh, this is cool. So they're now using a really interesting type of mouse model where you can label immune cells or any type of cell of interest with a color just by deleting or introducing like a gene. Here they actually have this special mouse model where you can turn on the expression of a red protein. It's called TD tomato or TDT. This protein will shine red when you take the tissue out of the mouse. And they have this only expressed by mast cells. So what they do is they can turn it on by injecting a chemical that then results in like the gene being expressed, which we won't get into. And then they take the tissue, they take the whole choroid plexus out of the mouse's brain, and they find little red dots on it. So they're going to compare a normal mouse. So this is a mouse without cancer. This is a mouse with like the cancer that doesn't cause swelling. And then this is a mouse that has the swelling that's really bad. And you can see like right away that there are way more red cells in the mouse that has more swelling. So they're just, again, kind of showing like, yep, there's lots more mast cells that are in this particular tumor. And for some reason, the ventricles are bigger. Okay, so now they've established that. Now they want to ask, what are these mast cells doing? So they know that mast cells will typically like degranulate and like release particles that then do things to the tissue. And so they use a marker for degranulation, and it's this green marker here. So all of these green speckles are the granules.
Yeah, my dick's full of Windex.
Cloven. Okay, so they found that the mast cells in our really aggressive tumor, so this is the mice that you give them the tumor, their ventricles swell up, and in these mice, they see a lot more granules being produced by mast cells. That's really weird. So then what they do is they're now looking at the cilia. Okay, so let's understand why they made this jump. So in cases of hydrocephaly or hydrocephalus, where the brain swelling occurs, scientists have noticed that the cilia on the choroid plexus, that tissue that's inside the ventricles, the cilia, or those little fingers, they actually disappear and they get messed up looking. And it's always correlated with this swelling. Now, this could really mean nothing, or there could be an actual purpose behind it. The cilia are really important, actually. They're an important part of the tissue for maybe helping in the regulation and production of CSF, because they're kind of like sensor cells too. So what they notice is that in the mice, so in normal mice, these are the mice with no tumors, they have nice little cilia. Now you're like, where the frick are these fingers? It's actually these little red things here. These are cilia. They're really small, but those little tufts are little cilia on the cells. And when they give them the tumor that's really aggressive, you can see that there's now no cilia present. No cilia. So something about the presence of these extra mast cells, maybe it's the cancer, maybe it's the increased CSF. This is all kind of happening, and there's also no cilia. So what does this all mean, right? So what they do, and I forgot what this mouse model is. Oh, wait, SASH. What is SASH? Hang on. Gotta go back. SASH mice are special mice that don't have any mast cells. So let me highlight this part. So they made mice that don't have mast cells, and they call them SASH because, it's an acronym. It's fun. So they genetically delete them. They get rid of the gene that helps transition cells into mast cells. They get rid of that gene, and then they lose all of the mast cells. And what they see in the mice with no mast cells, so let me just label that. No mast cells. Is they say beautiful little cilia. Here's the cilia again. They're present. So clearly something about the mast cells is causing the cilia to disappear. So maybe the mast cells are like directly destroying the cilia. Maybe the mast cells are turning off certain genes that are required for making cilia, but something's happening. And Ip has a great question. Are the mast cells doing something, or are they a symptom? This is a great question to ask. Like, maybe the loss of cilia happens and then we get more mast cells. Or maybe there are plenty of cilia, but then the mast cells come in to the ventricles and somehow get rid of the cilia. So we have to think about this both ways. This is just more images of like damaged cilia. Here's what normal cilia look like in this image here. Beautiful little fingers. And then this is what damaged cilia look like. Honestly, it all looks like spaghetti to me. So I don't find these images super helpful. I'm like, looks like a bunch of strings all stuck together. So I don't know. I like the fluorescent images better. Like this is more helpful. This is less helpful. All right, let's move on down to here. So now what they do is they actually move into cell culture. So what they've done is they've taken the choroid plexus out of a mouse and they've made like a cell line out of it. Actually, here they're using whole mounts, my bad. They're taking the choroid plexus out and growing it in like some media in like a little dish. Now the dish that they're growing it in, it has holes in it. And it's submerged in liquid. So there's holes though. So the liquid from down here can flow up and interact with the choroid plexus. And what they put in the bottom of the dish, these red cells, are the mast cells that they've taken out of the mice. So these red things are mast cells that they've taken out of the mice that had the tumors. So now they're separating everything from the brain and they're doing it in a dish so that they can have more control over the different variables. So when the mast cells produce their granules, the granules can float up and interact with the choroid plexus here. And so basically this assay is answering the question, do the mast cells have to directly touch the choroid plexus, or do the mast cells produce granules that then mess up the choroid plexus and like get rid of the cilia? So they're just seeing whether or not the mast cells actually have to touch the choroid plexus to cause the cilia to go away, if that makes sense. You wonder what they would find in your brain dish? Would you like me to throw some mast cells into your brain? Can I have a section of your brain freezes? So now they're looking at the choroid plexus in the dish. And so here's the control that we have located here. This is just like a normal choroid plexus, and it doesn't have any mast cells. It's just floating in the dish. We see beautiful little red cilia. Look at those beautiful fingers. They look like hot Cheetos. They're just like, so there they are. Then they go and they give them the mast cells. So this is plus mast cells. Now the mast cells are in the bottom of the dish, so the mast cells can't actually touch these cells, but their little granules can. And the presence of the granules produced by the mast cells is enough to make all of the cilia go away. We have like a little bit of red right here, but if you compare it to this other image, it's not as much. So the cilia are going away. Now what they do, which is super interesting, is they introduce a drug called DSCG that actually blocks the granules and prevents the granules from binding anything. This drug is here. When they add the drug, they see that the cilia grow back, or basically they're present. We don't know if they grew back or not because we don't know if they actually were ever gone to begin with. But we can see that when the granules are blocked, then we get nice cilia. So this tells us that the mast cells, they're making granules. The granules interact with the choroid plexus and make the cilia on the choroid plexus disappear. And we know that the cilia and kind of like the presence of cilia on the choroid plexus correlates with how severe hydrocephalus is or how much swelling happens. So if we don't have cilia, there's more swelling. This is something that has been observed before. And that's why they're using cilia now as sort of a proxy. So let's go back up to here. Basically, they're using the loss of cilia as a visual proxy or something that we can look at easily that will sort of indicate to us how severe the hydrocephalus is. Would the different fluid pressure in the dish affect the results? Oh, no. So this is the thing, right? So they've now kind of switched systems where they're in a dish. You don't have pressure in the dish. But what they're using is they're looking at loss of cilia as an indicator of whether or not there would be more swelling in the brain if we were looking back in the brain. Because they know that in the brain, in conditions where the brain has swelled up, the cilia are gone. And so they are kind of making an assumption, kind of a logical jump here, that, okay, if we lose cilia, that is also associated with brain swelling. I know that's not like the most perfect way to do things, but in order to kind of study things in a dish without pressure, they have to use some sort of proxy for swelling. And this proxy here is loss of cilia. So I know that's not like the most satisfying thing to do, but that's what they're doing. And they actually continue looking at cilia a lot. So down here, yeah, we're not going to look at this one. This is just another model. We don't really need to look at that. Let's go to the next figure. Okay. So now they're asking the question. So we've established that something about the granules made by the mast cells is making the choroid plexus lose its cilia. We know that loss of cilia is associated with more fluid production. So what is the compound that this mast cell is making? So what they do is they kind of look at the expression of a whole bunch of different proteins by the mast cell. This is really boring. They're looking at like really highly expressed proteins from mast cell. And they find, let's see, they find that tryptase, I don't think they show it here. Oh, they do. They find that this gene, TPSB2, this is the gene that encodes the tryptase protein. So this is the gene that makes tryptase. And we know that tryptase, they were actually staining it earlier because it's one of those proteins that mast cells release. So they see that the mast cells here are producing a ton of this tryptase. So automatically, usually if you're looking for like a main sort of culprit for the phenotype that you're seeing, you're going to look for the most expressed protein. Boring. They're looking for the trigger. Oh, you're right. I shouldn't undersell this. They do really cool things. They're basically looking at RNA expression again, and they find that the RNA that is for the TPSB2 gene is really overexpressed in the mast cells from these tumor-bearing mice. And then they're like, oh my god, TPSB2 is a tryptase, and we know that tryptases can have sort of these active effects, but they don't know what yet. So what they do is they're gonna actually, to study the function of this gene, they want to remove it, right? Sometimes if you want to see if a gene is responsible for something, you want to just completely cut it out of the mouse. So what they do here is they're using this system called the Krelok system. What that means is that you basically have these mice that are engineered to express these certain little cut sites in red that flank or surround the gene of interest that you want to cut out. Here it would be TPSB2 or tryptase. And then they cross those mice with mice that have this recombinase. It's a protein that cuts. And when they cross the two different mice, they get cutting of the gene, and that gene is gone forever. So the Cre cuts here at the LOX P sites, and then what you get is no more TPSB. So the minus means that it's knocked out. That gene is gone. So you now have special mice that have no tryptase. So you can now study the role of tryptase in vivo, which is really cool. So they then take these mice that don't have tryptase, and they give them the cancer. So here they're giving them the E0771 cancer, and they're giving them the type that makes the ventricles big to see what the role of tryptase was in ventricle size, and also in whether or not the cilia are gone or if the cilia are still there. Here we have these mice, the CPA3 TPSB2 knockout mice. These have no tryptase. And then they give the cancer to either a normal mouse here or the knockout mouse. And then they look at the presence, they look for the presence of cilia. Now, we already know that in normal mice, they lose the presence of cilia. So we don't see much of the red stuff. It's kind of all gone. And here's our like normal, completely normal mouse that has no cancer at all. And we see these nice tufts right here. So let me zoom in. You see the little tufts. When you give them the cancer, the tufts go away. And when you knock tryptase out, which again, tryptase is that protein produced by mast cells, you see the tufts are there. So now we have viable tufts. So this suggests that tryptase is somehow causing the choroid plexus cells to lose expression of cilia. And we know that loss of cilia is associated with more swelling. They do actually measure the amount of swelling that happens, because I'm sure if you're probably already wondering, well, did they actually confirm that the other symptoms were also rescued? So they do that. Well, maybe they don't. I think they do it in the next one. They do it in the next figure. They do it down here. They measure intracranial pressure. So they're looking now, okay, in these knockout mice, these are the ones that have no tryptase, which is maybe the trigger. It's what they think the trigger is. They see that the swelling goes down. So this is, again, the big, tall blue peak here. This is just a normal mouse with the tumor. So I'm going to just write plus tumor. And then the little orange thing right here, this is just a completely healthy mouse, which is always the baseline control that they compare everything to. And then they see that they're now looking at the CSF production, because remember, there was a ton of CSF production when you have that tumor that swells up the ventricles. But then when you knock out tryptase, you get less CSF production. And they also see a reduction in the neurological symptoms. So these mice are better. They're like healthier looking. And if we look at the red line here in the survival curve, we see that these tryptase knockout mice, so the ones with no tryptase, survive longer. So we just rescued the mice. We just made them survive better, which is really cool. Okay. And then, yeah. Okay, so that's all. We already looked at the cilia. We saw that, you know, in the knockout mice, they have their cilia back. Woohoo. They have less fluid. Woohoo. They now look at another protein. They look at this protein called NPFFR2 that is part of the cilia. So here, let's look at this normal mouse. The NPFR, it's basically like a neuropeptide receptor, and it's located as part of the cilia structure. And this neuropeptide receptor actually controls directly how much CSF is released from these epithelial cells. So, like, for example, if you lose the neuropeptide receptor, or there's like no signaling or whatever, actually, I think if you just lose it completely, you get more CSF leaking out. Yeah. And then you can see when you have the mice here that do not have cilia, so no cilia, these are the ones with that tumor that makes the ventricles swell, you can see that the neuropeptide receptor is completely gone, which I just said, when the neuropeptide receptor is gone, there's more CSF that's produced. And this kind of makes sense because we see more CSF being made. Okay. So now they're beginning to see like, okay, maybe this target, maybe the target, or like the reason there's more CSF being made is because tryptase is like somehow getting rid of the cilia. And when the cilia are gone, we're also losing this neuropeptide receptor NPFRR2. And that is triggering the epithelial cells to release more of the cerebral spinal fluid and cause the swelling. So this is kind of a good hint in terms of what the mechanism is. Yeah, so here to kind of show that, they do this experiment where I'm going to just stick to the visual right here, I think. No, here. So they're looking at CSF production. They're asking, okay, if we artificially get rid of this neuropeptide receptor, will we get more CSF production? And in these conditions right here, this is where they knock out the neuropeptide receptor. You can see there's an increase in the CSF. There's more CSF. So this kind of confirms that, okay, when we get rid of this, this neuropeptide receptor, more CSF is made. And that's actually in the, these are in the mice that also don't have tryptase. So they're kind of showing that even though you don't have tryptase, if you still manage to knock out that neuropeptide receptor, you'll still get more CSF, even though before we saw that when there's no tryptase present, the mice actually have less CSF. But that's because those mice still had that neuropeptide receptor, but then they went in and just got rid of the neuropeptide receptor artificially, and that reversed the effect. Okay. So then they're like asking, okay, well, where does the tryptase actually bind on these cells? It's great that we know that the mast cells are making the tryptase, but where do they actually interact with on the cell? But they're labeling the receptor for tryptase, which is PAR2. They knew this already. And they find that there's like this tiny little sliver of red right here. This is the PAR2 receptor. And it's located right on the part of the epithelium that is part of the choroid plexus. And they see that, okay, so the tryptase could definitely probably bind to this PAR2, but we have to confirm that. And I think they do a PAR2 knockout, but I'm not sure. Once they identify the receptor that tryptase from the mast cells is binding to, they're like, okay, what do we know is downstream of PAR2? So the way cell signaling kind of works is if we go back over to our little diagram page, let's say we have our cell. Let's put our nucleus back into our cell. And let's say we have PAR2 on the surface of the cell. So here is PAR2. And let's say, in this case, tryptase is binding to PAR2. The tryptase is from the mast cells. And then what happens is when the tryptase binds to this receptor, there will be some changes that occur in the structure of this receptor, and it will send a little signal down into the cell, which will then ultimately turn on some genes. And there's usually like one main gene that we call like a master, well, it's actually a transcription factor rather. So it actually activates a transcription factor, which is outside of the nucleus. And we'll call this the transcription factor. And when it gets activated, so when the cell, like some signaling happens, it gets activated, it goes into the nucleus and it turns on a bunch of genes. And then the cell will start making things or doing different functions. And so they wanted to find out, is there like a master transcription factor or something in the signaling pathway of PAR2 that gets turned on when tryptase binds that ultimately results in the loss of cilia. So here the output would be loss of cilia. And then we know that the loss of cilia also is resulting in more swelling of the brain. So that's the question they're kind of asking. And they actually identify, they know that there's this transcription factor called FOXJ1. And FOXJ1 is responsible for making cilia happy. It kind of keeps them alive and well. And so they're like, okay, well, let's see when we have the tumor that causes the swelling, do we also lose FOXJ1? So they can stain for this transcription factor using like a fluorescent antibody. So that's what they do here in red. So I'm going to change this to red. They look in normal cells. So FOXJ1, it ultimately goes into the nucleus. And they can see like the red is here in the nucleus. The blue things are nuclei. And then the red things are the FOXJ1 transcription factor. And then they look In the mice that were given the really bad tumors, and they see a lot less FOXJ1. So you can see like here, there's none. There's a little bit like here, a little bit here, but it's not as bright as in the normal mouse. And then when they knock out tryptase, which is the trigger that ultimately leads to this signaling pathway that could either turn on or turn off FOXJ1, they see that when tryptase is gone, they actually get more FOXJ1 signaling back. So now this tells them, okay, so when tryptase binds to part 2, right here, when this binding happens, it's actually like shutting off or removing FOXJ1. And then that is resulting in the loss of cilia and more swelling. So that's like the really complicated sort of pathway they're starting to establish here. Yeah, they find a way to knock out the FOXJ1 thing, the FOXJ1 transcription factor. So here is like a normal cell. And you can see here is a normal cilia, but when they delete FOXJ1, then they lose, like the cilia shrinks. Basically, it's small. Take a whole bunch of cells where they've knocked out FOXJ1. They see that the cilia are much shorter and less present. So they basically shrink, which is kind of fitting with what we're seeing in the mice that lose the cilia. Their cilia kind of shrink and start to disappear. And actually, when they make more FOXJ1, so instead of knocking it out, they add it back in, then they get really long cilia and they rescue the cilia length. So then the cilia go back. So basically, they find this transcription factor. It's really important for making cilia long, which ultimately regulate the CSF production. So there's less CSF production if the cilia are present. But if we lose FOXJ1, then we also lose cilia, and then we get more CSF and more swelling in the brain. So let's go. I think we're almost done. Now we're moving into humans. Thank God. Okay, so now they are like, okay, this was all true in mice, but can we actually see this phenomenon happening in humans as well? They take the human brain choroid plexus, and then they are looking for tryptase positive cells. Remember, tryptase positive cells is our label for mast cells. And they're basically just like, okay, we know that mast cells accumulate in mice with swollen ventricles, but does this also happen in humans? Do they also have mast cells in their ventricles? And so they're looking for mast cells in green. So here, and they actually find a couple cells. Oh my God, but it's very few. It's actually interesting. Here they only have like 3 mast cells in the human brain, which is still a lot. These are kind of a rare immune subset. So yeah, I think these tumors were from patients who had metastases, and then they are just sectioning them and looking to see if mast cells are there. And if the mast cells are making tryptase, in which case they stain for the tryptase directly, so we can see there's a bunch of it right here in these little green dots. And the tryptase, just for those of you just joining, is like the little molecule that binds to the cilia, or the little epithelial cells that have the cilia, and basically makes them stop expressing the cilia. This then dysregulates that cell and they like start producing the CSF, which, although I'm still a little unsure, like I don't know exactly if anyone knows why that happens, other than that the fact that there's this neuropeptide receptor that's kind of attached to the cilia that is responsible for making more or less CSF come out of these cells. So I think that is lost too when the tryptase binds. And so that's why the cells they go nuts and make tons of this fluid, and then the fluid fills up the brain. They take like all of these patient samples, and then they quantify the number of mast cells in like other parts of the brain versus in the choroid plexus. So they basically take the same brain and they look in the parenchyma, which is like the outer part of the brain, and then they take the choroid plexus part, which is the inner part of the brain where the ventricles are. And they're just seeing, like, in these brains, where are the most mast cells found? And they find that the most mast cells are found in the choroid plexus. And then they look at, they're now actually looking at brain scans. They're looking at brain scans from people who had no swelling. So this is no swelling as kind of a negative control. Here's the tumor, and you can see they have these beautiful, delicate shaped ventricles, just super small, super lovely. And then they look at patients that had communicating hydrocephalus. This is the hydrocephalus that is formed or that occurs through just the buildup of fluid. And you can see quite drastically that in this patient, their ventricles are really swollen, really big, really filled with liquid, kind of like blowing up a balloon with liquid. All right. And then they take all of these patients, so they kind of cluster the patients in groups. They have patients that have no brain tumors in gray, right here. And they're looking at the amount of tryptase, remember? So tryptase is released by the mast cells. They look for the number of amount of tryptase in the cerebral spinal fluid, and like the people with no brain tumors have very little. And then they look at people with tumors, but no swelling. So these people have tumors, but no swelling. They're just trying to draw a link between tryptase and swelling at this point in humans, just to make sure that it's kind of happening the same as in mice. And then they look at patients, and they also see that there's like very little tryptase here. Then they look at patients who have tumors, and they also have swelling. And the patients with the swelling have way more tryptase. So now this is kind of correlating with what they've seen in the mice. Then they also plot this where they measure hydrocephalus, so the amount of swelling, using something called Evans score. I don't really know what that means, but basically there's more swelling up here and less swelling at the bottom down here. And then they plot this against how much tryptase is in the CSF. And you can see out here, the more tryptase you have, the higher your Evans score. So the more swelling you have. So this is just kind of correlating those two data points. So they're like, let's block tryptase. Let's get rid of the swelling by using an inhibitor. Now, obviously, we can't just do this in humans right off the bat. So they do this back in their mouse model. So here, as a comparison, we have a normal untreated mouse. And they're again using a length of cilia, which are these little fun things here, these red fingers, as a proxy for whether or not there's swelling. Because remember, no cilia equals no swelling, or no cilia equals more swelling. I'm just going to write that up here for those of you who are just joining. No cilia equals more swelling, because you could think of like the proteins associated with the cilia as like the faucet that turns on the liquid that causes the swelling in the brain. So then they give these mice the tumor, and the tumor, as we know, this special tumor causes swelling. So this one causes swelling of the ventricles because the cerebral spinal fluid is filling it up like a balloon. And in this mouse with no treatment, we see the cilia are gone. We see very small little red stubs. They aren't beautiful red fingers like over here. And then they give a vehicle. Now, what is a vehicle? A vehicle is usually, so let's say that this BMS drug is resuspended in, I don't know, some like chemical compound. Let's say water, for example, just to make things simple. It's usually not water. It's usually some other chemical. What they have to do to make sure that the carrier of the drug, in this case, the water, doesn't have its own effects on the mouse, they have to do a vehicle-only control or a water-only control. So the vehicle is just the same solution that the drug is suspended in, but without the actual active compound. So it's a good control to have. So here you can see that, okay, we still don't really have cilia. We have like maybe something going on here, but again, not really. Then they add the drug. This drug, again, blocks tryptase, which is the thing that fucks up the cilia. And we see here that the cilia are beautiful. So they were actually able to rescue the loss of cilia by blocking tryptase. That's really great. They also do the scanning electron images. These ones are really crappy. I can't tell the difference between any of them. They all kind of look the same to me, so we're going to ignore that. What we're going to look at, though, is whether or not this fixed swelling. So remember those images from figure 1 so long ago, where they were looking at the amount of swelling, and the amount of swelling is how big that orange sac is. So here is a normal orange sac. These are the ventricles. And they're labeled orange. This is a mouse with a tumor. So this has the tumor. And when the tumor is present, which is this blue thing, by the way, it causes the ventricles to swell. And the reason we can tell they're swelling is because they get bigger and they actually measure that orange. They measure the volume of this structure and they see that it's greater. Then they give the vehicle control. Again, we should expect no therapeutic response. We should still expect to see swelling. We do. These ventricles are very, very, very big. Now we give the tryptase inhibitor and we see the ventricles are really small again. And let's look at our control. So here's our control. They're nice and small. And here's the treated ventricles. And these are also nice and small. So we've actually been able to prevent swelling by using this drug. This is really helpful. And here they just quantify all of that. So they're looking in the volume. They're looking at the volume of the ventricles. And they see, like, here's a normal brain. Usually the normal brain ventricles are 10 millimeters cubed. When you add the tumor that causes swelling, the ventricles expand 3 times as big to up to 30 millimeters cubed. This is massive if you're thinking about the scale of a brain. You do not want that much swelling in your brain. So again, this is why it's so dangerous to have this. But when they use the BMS drug that blocks tryptase, we see that the ventricle size shrinks back down to almost normal levels, which is really great. Hopefully that's making sense. Hopefully you guys can sort of appreciate how awesome this drug treatment is, and the fact that basically through all of the work they did in the other 5 figures, they were able to figure out what the druggable target is to prevent swelling in brain tumors. Amount of pressure to see if adding their BMS drug has reduced the amount of pressure in the brain. And indeed, they see a drop in the intracranial pressure. It's still higher than a normal brain, but it's definitely significantly lower than when they don't treat the tumor. And then they also look at, again, they look at CSF production. They do see a reduction in CSF, the cerebral spinal fluid, which would cause the swelling. They see a reduction in neurological symptoms, and they see an increase in mouse survival. So these mice are living up to 35 days. Now, if we remember from the first graph, mice were generally living up to 50 days. So this isn't back to normal necessarily, or back to, when I say normal, back to the mice that had just like normal tumors. These are more aggressive tumors that they've implanted in the mice that cause the swelling that they selected for. So it's still not like the greatest survival, but it's definitely better than the no treatment control, which is way down here. This is with no drug. They also look at cilia. This is in a human cell culture now, which we're not going to really look at. Basically, what they do is they make organoids. So they make little artificial choroid plexus from human choroid plexus stem cells, I'm guessing. I'm not really sure how they generated these organoids, but they're like little, they grow them in a dish, basically. So you don't have to like kill a human for this. You can grow the live choroid plexus in a dish, and then you can treat it with whatever you want. And here what they do is they look at just a normal one. So this is the normal choroid plexus cell. The red stuff is the cilia. So here's the cilia in red. Yeah, that's tubulin. Tubulin is just a component that makes up cilia. And then they add mast cells that they have activated with a drug called C4880. Now, somehow they know already from the literature that by adding this drug to mast cells, it makes the mast cells produce tryptase. So they add tryptase-producing mast cells to their human organoid. And when they do that, they lose the cilia. Glenn Kenobi, thank you so much for that cheer. That really brightens my soul. We're so close to the finish line. So they basically do a couple more things with the organoids where they use scanning electron microscopy. And they actually, this is really fun. If you've ever wanted to see a cilia up close, it looks like a really fun protrusion. It looks like a little stick that comes out of the cell. And then here you can see now it's a little nub when they add the mast cells. This is plus mast cells. And then they see that when they give the drug, that fun little cilia grows back. It looks like a little tail or a phallic object, if that's what you want to think about. So yeah, they ultimately found that you can indeed prevent the swelling because they identified the druggable target, which is tryptase. Tryptase removes the cilia, dysregulates the cilia, which then results in all this cerebral spinal fluid coming out, and that causes the swelling that we see in patients with hydrocephalus who have brain metastases. Now, some unanswered questions that I have actually, maybe you have them too, are over here. What triggers the mast cells to degranulate? Because if you think about it, why on earth? So something about that cancer line that they selected, that VM cancer line, it makes something, we don't know what, that causes the mast cells to degranulate and release tryptase. So that's one thing they didn't find out. What about that cancer is causing the mast cells to degranulate? The second thing we don't know is, why are mast cells even in the brain? Where did they come from? Where did they go? Okay, so we don't really know why the mast cells are even there. So that's something else they'd need to find out. And something I had a question about, and I thought they would do this experiment. Maybe they did, and they didn't show the figure, but why? Basically, do we lose FOXJ1? FOXJ1 is the thing that makes, basically it turns on the cilia, or you could think about it, it takes care of the cilia, makes sure they keep growing. So we found that tryptase actually shuts down FOXJ1, and that shuts down the production of the cilia. So I also was wondering, if you lose FOXJ1, do you actually lose that neuropeptide component of the cilia as well that regulates the amount of CSF flow? And I'm guessing like maybe they just assumed people would know that this neuropeptide receptor is a part of cilia. So if you lose the cilia, you also lose this, but they didn't state that directly. So I'm a little confused about that. But yeah, so anyways, the ultimate like big juicy takeaway though is that they found a way to like block tryptase. So we could take out tryptase, prevent it from actually binding its receptor here, and that way we prevent swelling and patients can recover happily, healthily, and hopefully live a great life. Yeah, I'm not going to really read the discussion because I kind of just said what my big takeaways were, and I'm kind of tired of reading. So I'm going to leave you guys with that. And hopefully y'all had an interesting time. Maybe you were sleeping. That's cool. Sleep is important. Honestly, if this was a time where you fell asleep, I am very happy because sleep is so important for your health. So if this was a good nap time, that's great too. If that were a machine, the cilia look like a tilt sensor. You know, if this is great because that you mentioned that, because cilia do function as a sensor. I don't know too much about cilia though, but I do know that they can like sense the environment. That's kind of why we have them. But I don't know what else they do really. So maybe if I knew more about cilia, I could give you a little bit better intake or like input about what I think is happening. But yeah, the whole process they went through is pretty fascinating, right? I think one of the things I wanted you guys to take away from this whole presentation of mine was just how many different things go into finding like basically not a cure, but finding like helping us pick a drug that we can use to alleviate a symptom, for example, in patients in like a real like in this case, when patients have brain mets and they have swelling, like how can we fix that? We've never really known how to fix that. But here they're saying, well, this is the target. If it's tryptase, if we can like either use what they used, which again, the BMS drug that they used, its side effect is that it causes hemorrhaging, and hemorrhaging is obviously really bad to have in your brain. So they were saying maybe don't use this drug in humans, but use something like it that functionally binds tryptase. Aren't cilia on the inside of the lungs that help clean the inside of the lungs? I think so. Cilia are basically everywhere. The cilia are inside of like your nose cells, for example. Cilia, one of their functions, you actually reminded me, Gear, is that they will like push, they're like one of the first physical barriers that bacteria, viruses, other like things you take up through your nose or into your lungs, they kind of push those things out with the mucus, and then the mucus comes out of your nose, right? The function of mucus coming out of your nose is actually to help get rid of some of like the virus and bacteria, and like cilia help push that kind of in a direction and also kind of act as like a guard. I wonder if I can find something a bit more specific about the other functions of cilia that we could look at. Oh, cilia also promote immune signaling. So they help immune cells communicate with each other. Well, I know that. So in like singular cells, cilia actually help them move. So if you're thinking about like, well, actually, I'm not really sure what type of cell. They're talking about single-celled organisms, like maybe bacteria or something. But let's go back to immune cells. People describe them as cellular antennae. So again, sensing the environment. Yeah, what else do they do? I'll have to look for that later, but they do many things. They're helpful for motility too. Living things are machines we don't have an operator's manual for. Atomic Quokka, welcome in, but we know what the programming language is. So the programming language for cells is the molecular cues that they send each other. And when I say molecular cues, this could be like different proteins that are released. And actually, we were talking about one, like tryptase is a protein. It binds to a receptor. That receptor goes and like causes this chain of events to occur inside of the cell, which ultimately result in that transcription factor, FOXJ1, being turned off. Now, there are other proteins that could actually turn FOXJ1 on potentially. That's just one example, but there's many like little like proteins and things in the cell that can turn on different genes, and then those genes will have other effects in the terms of the cell function. So if we can just control, so that's why, like, for example, let's say that there is a protein that causes a lot of inflammation. Sometimes you could block that protein with some drug that can like bind to the active site of the protein, or you can actually block the receptor that protein is supposed to bind to, and then it can't interact with it. So we do, once we like learn about where the proteins are binding, we can then go in and design other proteins or like synthetic drugs that can block those interaction sites. And that's how we control cell behavior to some extent. But then there's things like redundancies where maybe like you block this protein, but it has an effect we didn't account for. And, you know, we learn that through error and trial and error. But yeah, thank you so much for walking us through that article. It's rare to have someone trying to explain things to us normal people. Okay, just so you know, I was once a normal person. I think we all have the ability to learn how to read these papers. It really comes from just being in the environment for a long time and understanding like what the assays are, what the experiments are, because there's so much that goes into like, you know, one tumor experiment that you have to understand. And I've just been around this for a really long time. And I'll be quite honest, just last year, I think I would have struggled a bit to explain this paper to anyone. But I've been reading a lot of papers. Oh, I should turn my music down. That is loud. Whoops. But yeah, it takes a lot of time. And Quokka, you're asking, should I call you Atomic or Quokka? I'll call you Atomic because that's fun. My background is in immunology and microbiology. I actually should put something into my bio. I think I used to have it, and then I was like, oh, now I kind of sound snobby, so I'm just going to make it more fun. But I should really update my bio on my Twitch page. But yeah, basically I have a bachelor's in microbiology, and then I don't have it yet. I'm halfway through a PhD program in immunology, and I study cancer-related stuff. I thought your background was a bed and wall. Yeah, that is my background, bed and wall. Yeah, so right now I'm just, going through grad school and trying to become a better science communicator. So I very recently started forcing my Twitch channel people to listen to me explain random papers. This one was actually, I picked it out specifically for Styles Este, but he was feeling sick and had to go lay down. So the VOD will be for him. But he is really interested in mast cells and was curious if they have a role in cancer. Now, in terms of killing cancer cells, I haven't seen too many people studying that, but here we were looking at how mast cells are driving this symptom of cancer, which again, they didn't answer. Something about the tumor model they were using is like attracting or drawing in or somehow activating the mast cells, this special class of mast cells that are present in the brain and causing them to release this enzyme that then causes this brain swelling that we see. And so they're not direct, like the mast cells are more of a symptom rather than like a direct antagonist of the cancer. But I still thought it was really cool to kind of, shine a little light on mast cells because people in the cancer community don't think about them really at all. They're more important for allergy. So the way mast cells work, I should probably go pull my iPad back. Well, I won't. Basically, mast cells, they have a little receptor on them, not the ones that are found in the brain, but like normal mast cells that are found in your bloodstream or whatever. They have a receptor on them that binds to IgE, which is a type of antibody that B cells produce when there's allergy or when there's like an allergen, or maybe like, I don't know, some other, I don't know if IgE is produced in response to parasites too. There's certain types of parasites that trigger an IgE response as well. So the B cells, they get triggered, they make the IgE. The IgE floats through the bloodstream. It gets bound to the surface of the mast cell on their little special receptor. And then the receptor of the mast cell is binding to the constant region of the antibody, but the kind of the variable region of the antibody, which is the two-pronged part of the antibody. That's the part of the antibody that binds to targets. So when the mast cells binds with this two-pronged antibody to like a target allergen, that causes the mast cell to then degranulate. And the granules, one of those is histamine that we're aware of. So then we take like antihistamines to block the histamine. So when you have an allergic reaction, and you have to take an antihistamine, what you're doing is you're literally blocking mast cell granules, which are the histamines. So you're basically kind of replicating what they did in this paper, except not in the terms of cancer, but you're giving yourself a drug, and that drug then binds to the histamines, which have been released by the mast cells. So that's usually how people think about mast cells. Yeah, also unanswered. What were the mast cells trying to accomplish? So again, mast cells, they aren't really they don't have a consciousness. They're just responding to environmental cues. And I'm guessing there was a certain like protein cue that was either produced by that that special cancer that they selected for that then activated those mast cells. And those mast cells, I think what they did show in the later part of the paper is that there are a small portion of these special mast cells that just kind of live in the choroid plexus. But again, why are they even there? Why would we have these very bad cells? Not really bad, but why would we have them in our brain? They must be serving another function that through just like over time and evolution is actually beneficial for us. But what that is, we don't really know. We only know what what happens when they are unleashed in a bad way by cancer. So yeah, that's something that would be really cool to see a follow-up on. The way I was explained in trauma settings is swelling causes your brain to expand. If you expand something in a finite space, the skull, it'll find the weakest point and tries to squeeze and break through the magnum foramen. And I actually don't know what that is. And trying to squeeze your brain through a 1-inch hole. Oh, is that like here? Results in death, not to mention bruising the surface of the brain against the skull. Is it true also, Stike, that the swelling just kind of pinches or closes up blood vessels and you starve your brain of oxygen? That's kind of how I thought brain swelling resulted in death, but I don't really know. You have the training. I also have some food, so I'm just going to eat that because I'm like kind of hungry, and yeah. That's basically if your brain turns to mush. So in a trauma setting, the rapid onset of swelling usually outpaces that. The most common cause of death for brain hemorrhage or CSF leakage is the brainstem being squeezed down the magnum foramen. If it's compressed enough, it stops functioning. Brainstem is the part that deals with breathing and vital body functions. Okay, that seems to make sense. That makes sense. However, long term, the compression of the brain against the skull causes bruising, which does cause damage, how you'd think, pinching off of blood vessels and nerve endings and stuff and all the good stuff. So basically, rapid swelling can just result in death through, again, that mechanism where it's like pressing up on your brainstem, which then shuts down like important functions of the body. But if it's like slow swelling, then you get maybe eventual like pinching of the blood vessels and nerve endings and that. Magnum foramen is the hole in the base of the skull where your spine connects to the brain. Okay. That's super interesting, Stykes. Thank you for explaining that to me. I learned a lot from you just now. Yeah. Anyways, on my YouTube, I have two YouTube channels. One of them is designed for more silly things. That is my normal YouTube channel. And then I have a YouTube channel where I post VODs, and that includes the science talks. I was thinking about making a third science channel, but I just don't have time for that. So, yeah. I'm eating eggs, and honestly, I don't want to gross people out and turn you off from eggs, but eggs remind me of the texture of a brain. Just saying. Oh my god, that's spicy.